deletion. Okay. So when there are differences in single nucleotide, we call it substitutions. Okay. So of course, if you look at two sequence, never we have sequence totally the similar in size. Yeah. So we need to consider all this together. Yeah. So the pairwise alignment is actually to compare two sequences, whether it's DNA or protein, and there are lots of possible alignments. And whatever is it, the two sequences can always be aligned. The problem is whether the alignments make sense or not. So the sequence alignment has to be scored. So the scoring here is our major our emphasis today. Why you need to do the scoring and how we do the scoring. Okay. So the idea of global alignment is we try to fulfill the whole sequence. Okay. We try to end to end from the end of sequence 1 and sequence 2 to the end of sequence 1 and sequence 2. We do the alignment. Okay. We do not want to miss anybody in the whole sequence that you have. Yeah. So, but local alignment, this is called global alignment, but local alignment doesn't care the other sequence which are not really matching to each other. The local alignment only focus on a subset, a segment of the DNA sequence which is similar. Okay, so that's the idea. So this is alignment outcomes. If we do the global alignments, yes, for the above one, we have more matches. We get the whole sequence aligned together. Yeah, and then in order to make the whole sequence align together, we need to add up the insertion deletions. We need to add those who are not matching and those in the different size need to be added so that it becomes an insertion deletion. Okay. So in this case, the upper one, you see 7 matches, 3 mismatches, and then 4 insertion deletions. But if we do the local alignment, we only has lesser mismatch and insertion deletions. Okay. So that's the idea of the global and local alignments. So why we do the global alignments and why we do local alignment? So
Doctor,
yes, not moving.
the slide is not moving.
Where am I now?
Still, okay. Now, okay, alignment outcomes kan?
I think I don't want to put the full screen on. Always like okay. Right. So this is the comparisons. Why we do okay? We know that whenever we do the DNA sequence or amino acid sequence alignment, there are 2 methods. One is called global, one is called local. So global, we want to do the end-to-end alignment, but local, we're only interested on finding a specific segment. So a subset, a small fragments of the total size of the DNA or the amino acid, we call it local region, segment, local region, or whatever, with but with the highest similarity. Okay, so local alignment aim for aligning distantly related sequences, and global is for those who are closely related. So the application will be compare homologous genes with similar function in the global alignment, but the local is to identify short conserved regions into proteins. So the short conserved regions in proteins could be domains or motif. These two are actually the functional parts, functional regions of proteins, because proteins, we know that it is in 3D structure, so that in particular sequence, whenever it fall into the 3D structure, it become the functioning site. It could be the active site of an enzyme. It could be an interaction site with the target or whatever. So it could be a domains or motifs. We will talk about this later on, don't worry, in the next lecture. Yeah. So local will be fast and global will be slow lah. But what we are going to do next is we focus on local alignment. So we always say that we do BLAST, right? BLAST. BLAST is basic local alignment sequence 2. So BLAST, whenever we say BLAST, we know that we go to the gene bank, we put our DNA sequence there or amino sequence there, then we click the BLAST. Then the BLAST will come out in a list. You know that, oh, this is my gene, and this is similar to what sequence. Oh, then this is my sequence. I know that this is what gene. Okay, so then you will wonder why we need to understand global and local alignment here, because the local alignment is actually as it said, it is searching for local region with the highest similarity. So it doesn't uh, take into consideration the whole DNA sequence. So imagine that why we need to do so, because if we take up the whole DNA sequence to do searching, it will take up a lot of memory, a lot of computation times. Yeah. So we are interested in searching similar sequence, and therefore we only start off with those sequence in the segment of DNA to be similar in the database to the other sequences in the database. That's why we do the local alignment using BLAST. Okay, that is the application of local alignment for our daily life as a scientist in molecular genetics, or in bioinformatics or molecular biology or whatever. So we got the DNA sequence, we want to know it, we blast it. Yeah. Okay. So we will talk about a blast in details in next lecture. Yeah. This is still lecture number 2. So next lecture will be lecture number 3. We focus on blast. Okay. So the outcome of alignment should be optimal to maximize the number of matches and minimize the number of gaps, or adding gaps to reduce mismatches. Okay, this is actually a balance between two. One more gaps or less or more mismatch. Yeah. But if too many gaps, too many in order to fit up the mismatches, it is actually not biologically correct. So we need to have a scoring system. Yeah, the scoring system, we need to have a scoring scheme. So for those matches, we give it a positive value. For those mismatch, negative. For those uh, making up gaps, we need to give penalties. Okay, so the penalizing gaps forces alignment to have relatively few gaps. Okay, so you have no idea what is all this. Simply put, this is a balance. As I said, whatever sequence we throw inside the program, will give you something. But that something will be meaningful or not. Yeah. So that's why you need to have a scoring scheme. So what is this scoring scheme? It aims at providing a scale to measure the degree of similarity between two sequences and thus make it possible and quickly distinguish among the many different alignments that can be generated for any two sequences. So that means sequence matches could be anywhere. Yeah. So we need to ensure that which alignment are actually meaningful. So there are two separate elements here. First is to assign a value to pair of aligned residues, and second to assign penalties to gaps. So as I said, we need to do the scoring. So this is a scoring similarity. So only score aligned sequences. We focus on match and mismatch as well as gaps. Okay. But for protein, is it is simple for DNA sequence, but it's very complex for proteins. Okay, so we'll look at more examples later. So because amino acids have varying degree of similarity, we need to take the consideration that number of mutation to convert one to another, observed mutation frequency, and chemical similarity. Okay, so what does it mean here? This is the background of the substitution matrices, the PAM and PLOSM that I am going to talk about. So we know that from one amino acid to become another amino acid. So if you refer to the table of the genetic code, there is 64 genetic codes. Okay? But there are only 20 amino acids. So that 20 amino acids make up 64 codes. So you may be wonder how this is. So the genetic codes come from DNA kan, DNA, RTGC, 4. So genetic code is triplet. So it is 4 times 4 times 4, 64 combination. So 64 combination of nucleotide sequence in triplet form give rise to 20 amino acids, including the stop codon, 3 stop codon. That means there are 61 codons are actually coding for amino acids. All right. So there are a lot of redundancy here. Okay. So upon mutation happen in the DNA may not change the nucleotide, the amino acid sequence. Yeah, this is called the silent mutation. Okay. But if there are changing of amino acid from one amino acid to another, so there could need to have different mutations that change the amino acid sequence. Okay, so and then if the if we need more mutations, let's say the amino acid, the genetic code, we change one nucleotide, is not enough to change it into another amino acid. We need to have two mutations. So we need to have count the number of mutations to convert from one amino acid to another. And then in the real life, we got all the DNA. Let's say we look at one protein family of many individuals in the populations, as well as those species in the related of the related homologs. We want to see whether the mutation is frequent or not. Yeah, how frequent it is. So this is called the observed mutation frequencies. It could be from, let's say, amino acid A become amino acid B, could be quite frequent, 40%. But amino acid A become amino acid C, could be very rare, just 1%. Yeah. So we need to consider that in the real data we have for all the individuals, for many related homologs, then we come up with the observed mutation frequencies. So this is not a experiment data generated in the lab, but it's actually a combination of many data come up with these mutation frequencies. Also, we need to consider chemical similarity between the amino acids, like the polar, non-polar, and all those things. Okay. So the choice of matrix can strongly influence the outcome of the analysis. So still yet, we not go to the matrix yet. Okay, so this is an example of DNA scoring system. So mismatch could be 1, 0, match could be 1. Yeah, but in BLAST, match is 5, mismatch is -4. Okay, so different programs set differently lah. So, but the idea is the same. Match will give you more positive value, mismatch is negative, and penalty for gaps will be heavy, right? So we do a the so-called matrix is actually, if we refer back to your our degree, we we when we learn matrix, right? So we align two sequence together, two string of number together, and come up with the matrix, right? So the same way for this pairwise alignment, when we do alignment, we will see the identity matrix between sequence 1 and sequence 2. So imagine that the row, the row is first sequence, the column is the second sequence. So we try to the mismatching over there. Okay. So this is how to penalize the gaps lah. So the idea here is, if we do the alignment without considering the gaps, we only got, for the first example, the above one, we only got 4, score 4. But if we do alignment and try to do as many as possible, to align as many as possible, we got to insert gaps, which is the insertion deletion actually. And from that, we got 8 matches compared to the 4 matches above. But as I said, gaps must be penalized, because if you put too much of gaps in order just to make it cantik more matches, so actually this is not biologically relevant. So we need to have a balance here, and the balance here is actually, we have a very steep penalty on gap openings, right? So gap opening must, for sure you will get a 3, okay, the minus 3, and extension of the gap from additional 2, total 3 will be add up 0.2 here. So the total score of the alignment is 4.8, even though there are 8 matches, but because of this gap opening, it has a steep penalty, become 4.8. Yeah. So that's been the we are not really so fan of fancy of um, opening more gaps here. So we need to be careful here. All right. So later on we will have other things we call this e-value. E-value is another significant significance for alignment. So that all this matching or mismatching will have a statistical support. Right? It's not simply just mismatching a score, or I score this and that. No, we need to have such a statistic significance test. Okay. All right. So further on, now we focus on amino acid. Uh, so amino acids could be many overlaps of biochemical properties. It could be small, it could be large, it could be aliphatic, it could be hydrophobic, aromatic, charged, polar, and so on. And actually, this is not the end. There are also uh, um, so those in the same group are considered of the same properties. Okay, so this set of those are some properties will be called the similar amino acids in alignment. Okay, so let's say you've got arginine, and then compared to alanine. Okay, so this is totally two different amino acid. So in alignment, it could be just simply a mismatch. Yeah, but if you got an arginine here, and then for the reference sequence, you got arginine, your sequence you got histidine. So this is still highly similar amino acids. Yeah. And then how about if you got uh, look more here. So if you got a polar R, and then you got a charge E, right? This is still similar one. So how they got similar, highly similar. So this is need to be based on empirical data. As I said just now, we can't just say that uh, this amino acid is score how much, yeah, compared when it mutate to another amino acid. Cannot. We need to have empirical data to back up. So just now I mentioned that we need to have a lot of individual being sequenced. We got to know the amino acids. If also we also got to know a lot of sample individuals from those homologs, yeah, from different species. Now then we only know that for this particular gene, the protein, okay, so in the position, which one, the amino acids change to what amino acids, how is the frequency, and so on. Yeah. Then we come up with the empirical data background. Yeah. So eventually, after we have a lot of uh, protein sequence together, we found that the scientists come up with this relative mutability table. Okay, so it uses the alanine as the standard lah. So it means that in this table, those who love to mutate is this amino acid C. Amino acid C is what? Uh, Cysteine. Okay, and the relative mutability is 262.91. It love to mutate. Okay, and then who not really love to mutate is L. L is what? Um, L is leucine. Okay, so this is making up more complexity here. Just now you say you wonder what. Just now you say biochemical property. Now you say the relative mutability based on a lot of data, a lot of sequence here and there. Yeah, this is the empirical data. This is the background in relative. We found that simply put here, the table shown here is C love to mutate and L the least mutate one. Yeah. So this is to give you the idea. Different amino acids become another amino acids actually have different uh, what we call it probability. Okay. So that means from C become another amino acid in compared to A. So it could be have 2.6 times more frequent to mutate. Okay. So imagine 100 is 100%. 262 is 262%. That means when we do the comparison, the relative mutability, C to mutate to other amino acids is 2.6 times more compared to A. Okay, so I hope you understand the idea here. All right, so the penalty, the same thing now. Okay, then, okay. Uh, when we say uh, probability, okay, just now I actually I insert the idea of probability here. Okay, so the relative mutability is based on probability actually. So in order for us to know that whether the alignment score is really uh, statistically uh, meaningful or not, we need to have the probability study. Okay, so simply the simply idea here, I don't want to talk statistic because I don't even know what is statistic. But as I said, scientists especially those biologists when they become when they utilize the statistics to interpret biology and they find the meaning of biology based on statistics. I say I say that's the Superman because they could master both biology and statistics all together and apply it. Okay, so whatever is it, whenever we want to talk about whether this something happened just random or simply just random, or it is something happening real, yeah. So we need to have a statistic uh, backup. Okay. So we have put two models, random models and match models. So random models is totally not related, but it just simply match. Okay. So match model is sequence are related and that's why they match to each other, they align to each other. Okay. So the random model tells us that the sequence are not related at all, but simply random matching only. So it has no meaning here. But match model is because of the similarity due to homology, they match together. So in order to put this into statistic significant test, we need to tell we need to set the algorithm that the match model must be above the random model. Okay, so so we must be ensure this idea is the match model is actually really happening. It is not random. It is not simply by chance. Yeah, this is really something homology. Okay, that's the idea. Okay, so then you will wonder what is this substitution matrix in biology? Okay, and how it's related to evolution and homology. So as I said, mutation give us variation polymorphism. And the polymorphism we collected will give us some uh, uh, divergence. Eventually, there are homologs. Okay, so mutation gives us differences. So collection of differences will divide us into subgroupings, and this is called homology. Okay, those subgrouping are actually related to each other because although there are mismatches, there are mutations, still overall they are still similar. That's why this is homology. Okay, so uh, so how to put this idea into substitution matrix? Okay, so firstly look at this phylogenetic tree. Okay, phylogenetic tree is a best illustration of homology, evolution, and homology. So the sharing point is a common ancestor lah, the node. Okay, the node end of the. So when it divert into two, so that means there are two groupings, two subgroupings. So from the uh, original one, I change to G, C change to G, and so on. So you got two progenies, dua anak, okay? Yang lain sudah yang different in nucleotide amino acid sequence already. So when there are further mutation that cause the amino acids to change again, let's say in this case E change to D or A change to D, it will give us a subsequent new amino acid sequence in the next generation. Okay, so the same goes to the different side, the other side lah. So eventually, from a common ancestor, there are two progenies, and that two progenies are independently to produce another two progenies, and eventually you see there are a total of four differences from the same common ancestor. Okay, so you see in the between there are mutation, and mutation is recorded, and the mutation is is inherited. So during the inheritance of the previous mutation, new mutation also happen, and then it will keep inherited to the next progeny. And if there are new mutation, then the mutation will be collected, and so on and so on. So eventually there are a common ancestor, a nenek moyang, a nenek moyang, and nenek moyang, and so on. Okay. So this is the how to make this four DNA, four amino acid sequence together, and then we do the mismatch, the matrices. Okay. So the substitution matrix is calculated by observing the number of accepted mutation in the constructed phylogenetic trees. Okay, so don't care about the phylogenetic trees first. We will cover phylogenetic tree later, and then I don't know which lectures already. So the substitution matrix is calculated by observing the number of accepted mutations. So as I say, mutations are collected. Mutation happen. We carry the previous mutation from the ancestor. Then we also have new mutations. Then our anak will have our previous mutation from the ancestor and the new mutation from us. Okay. Then cucu kita will have another new mutations from the anak kita. So that's the idea. So put all this in mind, will make you understand what is this all these molecular genetics things about inheritance. And from where we develop a lot of things, from this idea, we develop a lot of things, matrices, calculations, statistics, and bioinformatic tools, and so on. Okay, so we need to calculate the probability lah. So the probability that mutates, then probability that A mutate to B. So we need to have this in mind. Yeah. So eventually we have the odd score, odd ratio. So don't worry, I'm not going to ask you to memorize all this and define what is QA and B. So the idea here is, as I just now mentioned, let's look at this. A positive value, the probability of those two residues being aligned is greater in the matched model than the random models. So this is the idea here. Just as I mentioned. Okay. So we need to ensure that the probability is actually the real one. Yeah. The match model is greater. The probability of match model is greater than the probability of random models. So that's in simple words is what we found is real. It's not simple by chance. Okay. All right. Eventually, we go to this POM scoring matrix. So last week we stopped somewhere here then. So all right. So I mentioned that for aligning amino acid sequences, we have different scoring metrics compared to the DNA. DNA alignment of DNA is very straightforward. So you got mismatch, you got a positive score, mismatch, a negative score, and gaps. So for aligning amino acid, also the same, but it's much more complex and need to take into consideration of the relative mutability, the table I showed just now, the chemical groupings of amino acids and its roles in the protein structures. So we need to have such a background empirical data first before we could do the scoring of amino acid. Okay. So Hoff et al. This is about 1970s. Yeah. So this is the crazy scientist. She got a lot of protein, 34 protein superfamilies of 71 probes of new homologus sequence. And then she got all this data on her hand. And then she want to know the mutation, estimate the mutation probability of each amino acid. That's mean from just now I mentioned that from 20 amino acids, from amino acid 1A convert to amino acid B, what is the probability? Amino acid A mutate to amino acid C, what is the probability? So this scientist has all the data on hand, the empirical data. That means she got all the amino acid sequence of 71 groups of 34 protein superfamilies. And then she construct phylogenetic trees and estimate mutation of each. So the estimation of mutation probability by using non-mutational history based on the evolution actually. Okay. So after taking all this evolution, mutation, and this empirical data together, so she come up with this PUM scoring matrix, which is points accepted mutation matrix. So that means a mutation is accepted if the new amino acid gives the same effect, has the same function to the whole protein. Also depend on how critical the amino acid is. So PUM square matrix always come with number, PUM 1, PUM 100, PUM 250. So 1 PUM means one accepted mutation per 100 residues. So from this, she construct the substitution matrices. So as I mentioned just now, so what is the probability of A? Amino acid A mutate to amino acid B. So what is the probability of mutation? Amino acid A mutates to amino acid C. And A, B, C are, B could be a similar amino acids, but C could be a totally different amino acids based on the biochemical properties. Okay, so that is different probability, but based on the empirical data, this could be counted. Okay, so eventually all this we make it into a scoring matrix. So this is a mutation matrix. Okay, so the mutation matrix here mean that look at the arginine versus alanine. It is just one. One is actually 1 over 10,000. The probability is very, very low. That's mean, okay? So assume that one is a point. Okay, this is PUM 1. Assume that over 100 amino acids, one mutation is accepted. So look at this highlighted one, the blue box and the red box. So you look at the title, I mean the title of the table. So the x-axis, the columns are original amino acid. Okay. So the y-axis, the rows are actually the new amino acids to replace the previous original one. So that means that in this case, for ASP, aspartic acid, become the glutamine. So you see the crossing point is 56. So this is simply just 0.56% of probability that ASP will be replaced or ASP will be mutated to GLU. That's the idea. Okay, so if we have this mutation matrix in our mind, then and this is the based on I keep emphasizing empirical data. So in this case, the empirical data is that 34 protein superfamily and 71 groups. Yeah, and based on 81 similarity share between all these amino acids. And then it come up with this table. So that means that, I repeat, for SP to convert to mutate into GLU, the percentile is just, the percentage is just 0.56%. Okay, so then how we score it? So of course, this is a very rigid mutation. We always relax the mutation into 25 mutation in 100 residues. That means the mutation rate is super high. And for scoring of the distantly related proteins. Okay, so if you don't understand what I say, it's okay. Let's look at the example. So this is a HB hemoglobin A and hemoglobin B is yeah. This is alpha and beta, yes. Hemoglobin alpha and hemoglobin beta, this orange brown box. So when we align two, okay, so for, if you recall the molecular genetic class, the last lecture we talked about globin genes, right? So the beta and the alpha globin genes are actually uh, from the same gene cluster. The gene cluster come from gene duplications. So that means alpha come from beta, or beta come from alpha, and then there are some mutation after this. Yeah. So there is some mismatches, and there are a lot of similarity here. So if it's so this is paradox here. So if these two uh, gene, this protein are aligned together, so we will look at the scoring scheme here. So G score 5, so S to N is similar, class is similar, but score 1, and so on. So you see a different amino acid, when they match with each other, the score could be different. Okay, so the scoring is based on the mutation matrix, right? So we look at further explanation here. Amino acids that make up the protein sequence have biochemical properties that influence their relative replaceability in an evolutionary scenario, meaning that um, whether the sequence to be, the amino acid to be replaced could have uh, biochemical properties that influence the functions and thus the replaceability. So what is all this? All right. The idea here is, so if there are mutation, it is accepted or not? Okay. So our perception of mutation is, if a mutation is bad, is really causing disease, make the people die, of course the mutation is not going to be accepted. But the mutation is accepted if the mutation is not really causing a lot of problem. It's not lethal, okay? It's not causing the progenies to die. So the mutation is accepted, okay? Uh, okay, many factors influence the probability of mutual substitution. Direct observation of actual substitution rate is often the best way of actual substitution rates. So that's why I keep saying the empirical data of by date of have this direct observation. So which amino acids change to which amino acids are all backed up by real data and come up with this matrix. Okay. So, but this palm, based on only one original data set that look at 85 identity. So that means when they have developed matrix, she filtered those amino acids, the protein sequences less than 85% will be removed, and only focus on those 85%, those protein sequences with at least 85% identity. Okay, so then after that, another group, Hanikoff and Hanikoff, so I forget the name here, come up with these blossom scoring matrices. So this is especially ideal for local alignments and at identify concert sequences based on similarity. So maybe you already feel what I'm talking about. So that's why I keep saying that the homology, the evolution uh, things at the beginning. So bear in mind that proteins functions based on stretches of amino acids which are conserved. Okay, what I mean here, let's say I take a protein, let's say amylase lah. Okay, so amylase in human and amylase in a cat, or even amylase in a yeast, are there are they different? Are they similar? So the idea here is why we call it amylase at the first place. Amylase, that means it is an enzyme that break down the amylose into apa nama maltose, right? So this means amylase in human, in a cat or in a yeast cell have the same function, right? So that's why it's called amylase. Amylase in human cat they are amylase because they share the same function to break down amylose to maltose. Yeah. So then you will be wonder, are amylase in human and cat and is the same? They are not the same. Yeah. In protein sequence, in DNA sequence, totally love this dissimilarity already. But in protein sequence, we could actually found stretches of amino acids in the whole protein. Let's say the whole protein 500 amino acid. There are stretches, maybe 10 or 15 amino acids in between, some here and there, a few stretches are conserved. Conserved here mean here that human cat yeast they are almost the same. Either is identical or highly similar. Okay. So these almost the same regions are actually the functional regions and make and tell us why they share the same functions. And that's why we name it as amylase. Even though the other amino acid sequence are not the same, but there are stretches of uh, highly similar sequence shared by these three species. Yeah. So because of this and highly similarity of this sharing of this similarity, we call it conserved, right? So this region is called it as conserved regions. And the conserved regions actually give us the similar functions of the protein. In this case, amylase lah. Okay. So this blossom is actually for local alignments. M at identify conserved regions sequences based on similarity. So recall that just now I said that the Tehoff matrix focus on those uh, protein sequence more than 85% similarity. More than 85% is actually very highly similar already. Yeah. So don't you ask, so how about the less similar one, those lesser than 85% similarity? So that's why this Hanikoff group of people, group of researchers, they come up with these uh, complementary matrices for um, less similar sequences. Yeah. So for those less similar sequences, still there are a lot of sequences are not similar. But as I said, because of similar in function, they should have conserved regions which share the similarity of the amino acid sequences. Yeah. In order to fish up these similar sequences, these structural similar sequences, we need to do the local alignment. So as this shown in this figure, there are structures of similar sequence in different species. Let's say one bar represent one species. So eventually, if we do the alignment, we will find out this is actually the conserved region. Everybody say similar sequence here. Okay. So in words is ideal for local alignment and identify conserved sequences based on similarity. So it's collect a set of multiple alignments and find the block. So this block, as shown here, you know that the sequence in white are the the non-similar sequences. So we only interested to find the sequences which are highly similar. So eventually it becomes blocks here. Okay. So the blocks here is actually called uncapped local alignments. So there should have no gaps here, a block. So based on a cluster of blocks of sequence that share at least a certain percent identity, n is, we will settle n later, count the occurrence of all pairs of amino acids derived from alignments of domain of distantly related proteins. So just now the pump is for closely related sequence proteins, but the blossom is for distantly related proteins. So for those distantly related protein amino acids, we have different metrics, okay, to say it similar way. So in the day health model, the scoring value are derived from protein sequences with at least 85% identity. So day health is actually the PALM model. So so this honeycomb and honeycomb, this is actually the blossom. Therefore, develop scoring matrices based on alignment of more diverse sequences. So more diverse, obviously diverse, that means you know that they are less closely related, they are more distantly related, and calculate the number of occurrence of each pair of amino acids. So similarly, this handicap group, they use multiple empirical data to calculate the metrics. So eventually, Blossom 62 is used as a standard for gap alignment. So 62% here, that means 62% of similarity. Okay. So this is a 62 substitution, Blossom 62 substitution matrices. So what do I mean here? So this is actually the scoring matrices. So if we want to do the protein alignment, we do the scoring, right? So for the scoring, when we align two amino acids, so based on Blossom 62, so if C, let's say in this example, map with P, so you got a score of -1, and so on. So let's say if C map to W, you got 3, score positive 3. Okay, yeah. So similarly, when we have all this uh, substitution, we put it into the statistic, we'll calculate the odd ratios and give us the number. Okay, so this is a scoring matrices lah. So C to C9 and then it's actually the same as above. Okay, so what is the dissimilarity between these two? As I said, PALM is for highly similar protein sequences. So if you want to do alignment for amino protein which is highly related, closely related, we use palm. If we use blossom for distantly related. But of course, this is not just a fix here. So, the why we use blossom, because general blossom is better than palm for local similarity search. So what I said just now is actually the scientists also developed the palm and blossom for less divergent and more divergent. Less divergent means more highly similar amino acids, and more divergent is uh, less similar amino acids sequences. So all these are for what? For the same purpose lah. So eventually, using the same metrics, they develop for less divergent and more divergent. So that's why they has different numbering here. So I say 1%, palm one is one is accepted for out of 100 amino acids. Blossom 62 is 62% is accepted, and so on. So of course the metrics will be different score lah. Okay, the idea here is different metrics will have different scores. Okay, so then eventually this seems like these two are overlapping and sounds redundant. So if I got palm one, blossom 80 are still the same. It sounds like that, right? So what is the difference here? Okay, so usually palm 120 and blossom 62 for general use lah. For palm 60 and blossom 80 for close relations, palm 250 and blossom 45 for distant relations. This usually go to opposite lah. Okay, the numbering. So just remember that. So, but still, blossom is better than palm for local similarity search because lower palm metrics find short local alignments, but higher palm metrics find longer but weaker local alignments. So usually use blossom season for database searching lah. So when comparing closely related proteins, should use lower palm or higher blossom, and vice versa. Okay, so the idea here is blossom is better for local similarity search. Okay, just ignore this slide. This is actually a raw slide. Okay, so if we have this bear in mind, then we know that what we are going to do is alignment using BLAST. As I said, BLAST is basic local alignment search sequence tool. So the name already tell you this is a local alignment. Yeah. So local alignment is very useful when we want to do the database search. Yeah. So then you can wonder why I'm talking this. Okay, very simple. Now you got a DNA protein sequence, amino acid sequence. So you want to search your amino acid sequence over a database. You want to know what is this amino acid sequence. And let's say you want to search an amylase, okay? But in the gene bank database, the amylase could be more than 10,000 or 20,000 amino acid deposited there. Yeah. So gene bank is a database, everybody can put their sequence. Yeah. So let's say the whole world, the people working on human, yeast, cat, dogs, whatever, they do the amylase, and then they got the amino acid sequence of amylase they put in the gene bank. So eventually there are more than 20,000 of amylase sequences in gene bank. Yeah. So that is not standardized one. Yeah. Some people may put the whole, let's say the amino acids, the amylase got 500 amino acids as a complete protein. So some scientists just focus on 100 to 200 amino acids. Some scientists just focus on the first 300. Some scientists the last 300, let's say. So the sequence deposited in the gene bank are actually quite heterologous. Yeah, everybody can deposit on it. Yeah. So when we do the searching, and don't forget these sequences could be derived from different species, could be very distantly related or very closely related species. So now you've got a sequence. You want to search over a database, we have 20,000 K, 20,000 sequences of different size, different length, different species, and so on and so on. Yeah. So if we do the searching like this, you could take years, take for how many hours in order to get a, a matching search. Okay, and that's why we need to do the local similarity search. Yeah. So we just use some segments of the DNA, your sequence, and then do a quick search in the database. From the quick search, it will shortlisted some potential one and further complete the searching. Yeah. So this is called the blast. And the idea here is this could be achieved when you got one amino acid sequence search over 20,000 amino acid sequence in a database. Yeah, this could achieve in just simply 1 minute. Yeah. So if we do global alignment, you have to ensure that everybody are matched end to end. I don't know how long it will take. Yeah, maybe a few days. Yeah. So that's why BLAST is actually a practical program for similarity search using local alignment. And the local alignment is just simply, it's not going to say that don't misunderstand that it just focus on one or two segments of your sequence and do the search. No, it's actually do the complete search, but it breaks down the sequence into multiple hits to search a database. So we will talk about this later on, so don't worry. So the BLAST will be used for similarity search of a query. Query here is your sequence lah. Well, whatever DNA or amino acid sequence you have, you put into the search machine. So this is called a query. Okay, so those match to your query is called subject, right? So use FASTA as a database search algorithm. Ignore this. This is not correct. So designed to accept gaps, generation of gaps alignments, task sensitive, weak but biologically relevant sequence similarity. So we keep focusing the same thing, biologically relevant sequence similarity. Yeah. So we need to find lah. So if we do search of high similarity, it's not really it's an easy job lah. Okay, compared to finding those weak but homologically evolutionary relevant similarity sequences. Okay, so the extension steps take 90%. Okay, so use dynamic programming to extend the central pair of aligned sequence in both directions. So we will talk about this later. Not only find the single best segment pair, but also other local alternative pairs. All right. So don't worry about this. We'll talk about the blast next. So this is actually this is lecture number 2. So I proceed to lecture number 3. So which is the blasting. So before that, would you like to have a break or would you like to ask some questions? Okay, also the. All right, I keep talking about all these things. Actually, if you read back, if you do the revision, you will find that actually the things are quite, I mean, the logic behind is homology, evolution. Yeah. Then how to score it. Yeah. Okay. So you will never learn this in any class, any subject, molecular biology, whatever. genetics, but you only learn so in bioinformatics. Because bioinformatics, as I say, uh, it's not a tools, it's not a techniques, it's not a analysis programs. Bioinformatics is actually a discipline. Yeah, you need to consider all this together and also use statistic, use computer, use database, and so on to give us the idea, the interpretation of the results. Okay. Oh, next. Okay, this is number 3, lecture number 3. Yeah. So we are going to use plus today and the concept plus. Okay, plus I believe you have done it before. As I, for my molecular genetic, the first practical, the online practical is plus, right? So you do that for molecular genetics uh, to solve a problem. I mean, giving you a task, you solve the task. Giving you this is a sequence, you call it in the lab. So what is this? Then you do the plus, and then you do the phylogenetic trees, right? So in bioinformatics, we we talk about the logic behind. Okay. Uh, okay. Can you see that slide then? The mutation and homology and evolution slide. So the same thing, the same slide actually, the same figure. So the idea here is random changes in DNA produce variation among individuals sharing common ancestor. Over time, a variant is fixed to a species adapting to a condition. Eventually, it's evolution. Bear in mind that this is the backbone of molecular genetics and bioinformatics. Okay, so bioinformatics, we have to use our data and search in database. Yeah. So public database usually are public database. Yeah. There are public data available in public databases. Yeah. So that's why I mentioned that let's say you got a amylase, there are many people are looking at amylase but on different species on or look at different portion of the protein amylase protein. So, but they just upload the findings, the amino acid sequence or even DNA sequence to the gene bank. On gene bank will be housing, storing all these DNAs amino acid sequence for for your references. Okay, so it's house all sort of DNA or amino acid sequences, structural information for different individual or different species, exposed to different conditions, tested with different parameters, and so on and so on and so on. Okay, simply put, there are public data in public databases. Okay, so want to know about your sequence, search it over the public databases. Okay, that's the idea. And once you got search the one that you want to look for, so you could just download the sequence, yeah, for free. So usually we will look at this for both DNA or you got amino acid sequence, you use the gene bank. Gene bank is the most commonly used one. Swiss prot is actually a protein database, but they are much more complex, much more informations. Okay, OMIM is actually a nucleotide sequence database, but it is a database that contain all, it has filtered all those genes and variations that cause diseases. Okay, the catalog of human genes and associated diseases. Yeah, right. So there are also database lah. Yeah, also database, but don't worry about this database. We focus on our work. Okay, we want to know that you got a DNA sequence or protein sequence. What should you do next? Okay, so we focus on the gene bank especially. So if we do the sequence search, we use BLAST. BLAST is actually a program installed incorporated in the gene bank search machine. Okay, so this is an outlaw of the BLAST. I believe you know it before. So it is a very important online tool for similarity searching of query sequences over gene bank for both DNA and protein. So it is a rapid heuristic algorithm allowing large database to be searched using ordinary computer. So algorithm is the, okay, how to define algorithm? Algorithm is actually the way of working, okay? How the logic behind. So these two, the searching, the alignment and similarity search tools, how they work. So how they works is actually called algorithms, okay? So heuristic is somewhat more computer things, computer statistic things. So I will give you the idea in the next slides. For gene protein identification analysis for region similarity. Okay, so I'll keep talking that we want to look for similarity, similarity, similarity. Now you can do it on the gene bank database. Plus got a lot of programs lah, but we usually focus on plus N and plus P. Plus N is the most common one. We search DNA sequence. The plus P is we search amino acid sequence. The other plus is not really quite different, difficult to use, I may say. So it's an advanced class for plus X, T plus N. So we just let you know the existence of these extra tools. So it is not only you can plus the protein to only protein, you could actually plus the protein to amino acid, to nucleotides, or in a reverse way. You could plus nucleotide to a protein database, also can. So, but plus N and plus P, you only plus nucleotide and protein respectively. Okay, we focus on plus N and plus P here. So how plus is developed? It's actually a local alignment. Yeah, I keep saying local alignment. Algorithms that optimize searching of local similarity. As I say, the amylase as an example, even though from different species, but it is called amylase because it has the same functions. Yeah, the same function come from the similar, highly similar segment of the amino acid that give the function to the amylase. Yeah. So this segment of similarity between crossing species is called local similarity. Yeah, or simply called conserved region. Yeah, among the species we call conserved regions, but when we do a search, we look for this conserved region and we call it as local similarity. Okay, placid or locally maximal segment pairs, the MSP, and we give a cutoff value score of S. So plus is also not simply Chinese that it must be a score, cutoff score. So the cost of alignment is a measure of similarity based on well-defined mutation scores. So well-defined mutation score is actually the mutation matrix, okay? The substitution matrix. Mutation matrix, substitution matrix is the same. So each search will screen through every sequence deposited in the database via pairwise alignment. Okay, just now I already said that you got one sequence, you search over 20,000 sequences in the gene bank. Yeah, so we need to, but it could be finished in just 1 minute. Yeah, how come it could be so fast? Because we use BLAST, we use local alignment, right? So the BLAST actually developed by this group of scientists. It was first introduced in 1990, and then it further refined in 1997. There are also a new further refinement, but we focus on the most basic two. Okay, so as we mentioned, gaps are actually good if you introduce gaps to get a better similarity scoring for distantly related sequences. But unfortunately, the the blast in the initial development is not for gap, yeah, not for aligning those sequences with gaps. Yeah, if you have problem to do so. Yeah. So then 1997, the gap blast was introduced. Okay. And then what we are doing now in the public in Jinpeng when we do blast is actually the even newer version of this blast lah. But it already incorporated all these gap things. Okay. So the idea of pairwise alignment, just now I said, now you got one sequence, you screen 20,000 sequences in Jinpeng. Yeah. So it is not simply just the screening is actually one by one. Yeah, but it could be very, very fast. The one by one screening, that means is I compare my sequence to the sequence number one in the Jinpeng, then sequence number two, then sequence number three, and so on and so on. Yeah. So whenever comparing two sequences together, this is called pairwise alignment. Okay. And then we find the best alignment based on scores. And those nucleotide or amino acids on the same diagonal line are aligned. So in this case, so you got two sequences here, sequence one, sequence two, and this is the, once we do the alignment, we got a possible alignment here. So you see there are just three matches and there are many mismatches in between. Yeah. But if it draw into a table, into a graph, and then we lay down the sequence one and sequence two in two axes, axis X and Y, from comparing the first nucleotide to the end. Okay. From first nucleotide to the end, and we only find three matches. Okay, so those on the same diagonal line are those aligned one. You see a T and the T. So there are one, two, three, four mismatches in between. Okay, this is the idea of pairwise alignment. So why we do local alignment? So, global similarities algorithms optimize the overall alignment of two sequences, which may include large stretch of low similarity. Local similarity alignment search on only relatively conserved sequence and single comparison may use several distinct subcellular unconserved region and do not connect to the measure similarity. So talking all this, as I say just now, verbally, yeah. So if I just look at this, you will never understand. It enables it to screen through all sequences in your database speed. As I said just now, 1/20,000, it could be finished in 1 minute. Yeah. So this is a speed. We want to the local alignment because we use it for speed. Okay. Identification of point mutation and identification of homology search identify small conserved regions. But still, we need to preserve the sensitivity. Even though it's fast doing local alignment, yeah, we still need to preserve. We are looking for differences. We must ensure that those differences must be searched as well. Okay, the sensitivity. So doing blast, we want to have the speed as well as the sensitivity. So this is the idea of doing local alignment of blast. Okay, so what do you get? So let's say we search a sickle cell anemia patient, beta hemoglobin amino acids. So in the Jinpeng, we got this amino acids, which is about 147 amino acids. Okay, so if we search over the Jinpeng, okay, we can find the amino acid sequence of any gene in Jinpeng. Just key in by Google search in the database. We can find the one that you want to look at. So when we do the blast in the Jinpeng as well, you will find some further sequences that you would like to look at Yeah. So in this case, um, the one in this reference sequence is actually the normal human hemoglobin. Yeah. But those search result, why I want to look at this sickle cell anemia punya beta hemoglobin. Yeah. So when we do the comparison of search of this normal human to your sample, which is from the sickle cell patients, sickle cell anemia patients, then we find that there are two mutations here. Okay, so there are matrices here. There are what we call it uh, statistic here. You how many bits lah, expect value, the e value, how many identity lah, how many are positive lah, how many gaps lah. All these are listed down here. So e value is statistic to tell us that this is significant. This is based on model match model. Okay, this is totally uh, significant. Okay, so the identity here is of 147. There are 145 identities. Identity means identical. If V is V, yeah. Positive is those who are similar or highly similar sequences. Yeah, they are from the same group of amino acids lah. And those who are totally got nothing is actually the mismatches lah. And no gaps here. Okay. So another function is to homology search is plus lah. The plus function is homology search. So myoglobin and hemoglobin share about 80% of sequence identity, the green one. But about 85% of sequence similarity, both green and yellow one. Okay, so you see the green one are identical. So if WW lah, so the yellow is different amino acid. You can see. Okay. How do BLAST work? So BLAST attempt to find short fragments of query sequence, that's your sequence, that aligns perfectly with a fragment of a subject sequence, the hit found in database. Use the dynamic programming plus heuristic method to extend hits so as to allow gaps in the resulting alignments, but it is slow. Okay, so BLAST is a local element. Local element look at similarity search of short fragments, okay, out of your sequence. Yeah. Then it also allows gaps to happen. So what happened when we do BLAST? There are three steps in the BLAST, three major steps. Okay. So the first step is compiling list of high scoring words, and then scan the database for hits. Lastly, extending hits with score and test of significance. All right. What to do? So now this is an example. Okay. Given you have a amino acid sequence here, this example SWV, SQAS, FTPPGIM. You want to search over the gene bank using plus P in this case plus P. Okay. And how the algorithm of BLAST work. So firstly, if you break down this sequence into overlapping segments, we call it words. So usually for protein, we have three amino acids in one words. Okay, so list of words is n minus w plus 1. That means if your amino acids got if your amino acids got 20 nucleotides, you will have 20 minus 3 plus 1. So 18 words, overlapping words. So the overlaps is like this. You see, it breaks down into overlapping words by firstly SWV, then WVS, and VSQ overlappings. Okay. Then each of these words will be used to search the database. So this is called the scanning the database for hits. But how? Which one is used? I mean, from 120, from one amino acid sequence, we break down into words, overlapping words. Then we screen through the database. Okay, but which words actually working? Yeah. So whether it's working or not, I mean here we need to have a threshold to say that, okay, this works when screened through the database, they are matching and how they score. Okay, the matching is based on score. So the score is actually the threshold value. So those score lower than a certain threshold, those words with score lower than certain threshold will be removed for further work lah. Okay, so in this case, you got a list of from this amino acids SWV, SQ, and so on. We got a list of words. Okay, then we do the log. Then okay, then we compile the list of work into. Oh, sorry. So we have to ensure that that words shortlisted one with the over the t value, the threshold. Usually it's 11 or 13. Okay. So calculate the score T for each word that align with sequence database using a subdivision matrix. Yes. So now the subdivision matrix come in, the palm or blossom. So in plus, we use blossom substitute. Okay. Discuss whose T value is below a neighborhood score threshold. T is actually called neighborhood score threshold. So if we have sets 11, that means those below 11 will be removed for further analysis. Okay. Then all these words that screen through the T threshold will be compiled and do the further analysis. What the further analysis here is actually to extend alignment. So let's say just now we got a SMV here. Okay, we will search through one word will be searched through the database. Okay, then we find the matching. So next step, then we will do the extension. So extension from both ends. So is this case SMW from S side before S, we'll try to extend the heat and so on. So the extension will, when a word align, extend it as the seed. So this word, when it's aligned to the database, will be used as the seed. So from the seed, it start the extension. So don't worry about these words, this seed, what that. So this gives you the idea only how the thing works, and that give it a name. Okay. The alignment is extended in both directions until the alignment score decreases in value. So when score based on blossoms drops more than drop off value X, hit or best score is found and extension stop. So that means you will keep going the alignment until the score drop. Okay. So the score is actually the blossom based on the scores, blossom matrices. Yeah. So don't worry, if you don't understand, we'll do practicals. But practical will be uploaded next Monday. I need to prepare some more and fine tune something before I could upload. So now you this is the idea, but later on you do the practical, you know what I'm saying. So the practical is actually more on hands-on. You are trying to manually doing the mapping. Yeah. So when you do the manual mapping using your hand and eyes, then you understand what I said based on this blossom and so on. So the gap extension is actually the key point in the class. So because it's in order to reach its speeds for screening through the database, they need to do the breaking down of the amino acids into words, right? But the words align back to the database, it must achieve the threshold. And then after it achieves the threshold, it will be extended for the map alignment. So, but this is for gap and gap. In order for the gap to be calculated together, so the second gap plus developed in 1997 used this two hit method. So the idea here is uh, those who are on the same diagonal line. So if you recall this, relate this to slide number 9, this diagonal line. So imagine that this graph, this axis extended long. So there are another pair of words that align on the same diagonal line. Yeah. So that will be used for extension. So this is called the two hit methods. Okay. So this is from the original paper, this figure from 1997 original paper. So when there are hits, they're on the same diagonal line, they will be used for subsequent uh, gap alignments. And this pair, we call it high scoring segment pairs. Yeah. And then a high scoring pair is a maximum segment pair. Um, so on the same, within a distance, but on the same diagonal line. That means they are actually aligning on the same DNA sequence, not by chance. This is by uh, how I say it. So it's okay. When we do the practical, I will show you more information. Okay. So this is a way that they make it the gaps to be covered. Okay, and make it much more effective also. It makes the alignment is about 2.3 times, 2.3 times faster. The gap alignment is much more faster than the original plus alignment. Yeah. So eventually they will generate the alignment and calculate the statistics. So eventually the query, your sequence, you get a subject. Yeah. The numbering is the first amino acid 1 and the amino acid 393 for the subject. It start the alignment there. And then you'll find these regions. Okay. So those the line in the middle. You see query one, then a blank, a subject. But if you look at the alignment, the query amino acid sequence, then this is plus and the alphabet, and then the subject amino acids. So those in the middle, the line in the middle is actually the showing you the identity. So if everybody is, example here, everybody is G, then it's identical G, identity G. Plus means there are similarity in amino acids. So recall that biochemical properties lah. Those who totally has blank is totally a match. Okay. So eventually you'll find patches of similarity here and there. So of course we need to have the statistic to support. So in class we call it E value. So the significance of a matching sequence we score as retrieved in blast search is expressed as the E value, expect value, E value. Is defined as the expected number of high scoring segment pairs we score at least S given a random query sequence of len M and a random database sequence of len N. What is this? Don't worry. So the idea here is it is significant. So what does it mean here? So if the E value is equal to 10, what is that converted into words that human can understand? So if E value is 10, then that means there are 10 aligned MSP. What's the MSP already? The high scoring scheme pair. Um, uh, uh, so MSP is actually in the original blast is called MSP when the word is matched to a amino acid sequence is called MSP above the threshold. But in the gap blast in 1997, the new development is called high scoring segment pairs. Okay, so the MSC is the maximal segment pair. This means it's an uncapped local alignment that is locally optimal. This means it is when you do a alignment, your query sequence map to the sequence in the database with above the scoring threshold. Okay, so in this e-value 10, assume that there are 10 MSP maximum segment pair with score more than X can just happen by chance. So it is by chance 10. So by chance is not a good thing. We know that we are looking for homology. Homology is not by chance. This is evolution. This is true happening in real life. It's not simply by chance. This is homology and inherited. So if you got 10 MSP by chance, searching those is totally not significant at all. Okay. But if your searching is no chance, it's less than zero, much uh, deeper, less than zero. Let's say this power is minus 10, means that it's much more unlikely that random chance lead to this current alignment. So, so that's why in the last lecture and this lecture, I use the homology of the phylogenetic tree because we keep saying that all these are homology, the matrix, the amino acid substitution matrix, and even after this, how we test it in a significant statistics. Yeah. So E value is actually to tell that um, whether this is simply random or not. So I repeat that E value 10 is there are 10 MSP, just happen by chance. But if E value minus power of 10, that means there are almost no MSP by chance to give you the current alignment. Okay, so what is the rule of thumb? So if there is significant homology, E value must be less than power of 10, power of minus 4. If is some social things is between minus 2 and power of minus 4. Or if your E value is more than minus power of minus 2, so this is not significant at all. The rule of thumb. Right? So look at the alignment defined for this hemoglobin, sickle cell hemoglobin. So you could see that the E value is 4 E minus of power of power of minus 101. This is totally significant. Okay. So this is the idea here. But the E value is actually a function of S value and the database size. Yeah, just now I mentioned that you got 20,000 in the 20,000 amylase sequence in database. Yeah. So it is a function of your match to the database. Yeah. So That's why the idea of randomness. So if in the database got 20,000, by chance, you still got high E value. That means this is not a real significant matching. Yeah. But what you got it is by chance is no way, no chance. Yeah. And that means this is the real things. Okay. So that's the idea. Yeah. So okay, look at the syllabus. Actually, we need to cover three major things. First thing is the sequence alignment, whether it's called multiple, global, local, multiple, pairwise, blast. All these are fall under the sequence alignment. Secondly is the phylogenetics. Then the protein structure, DNA, nucleotide, and protein structure. These three major things. Okay, so the lecture 2 and 3 cover the alignments already. Yeah, so only that we could have the practicals for these, as I said, three major subjects that we want to cover the alignment, including the global, local, pairwise, blast search, sequence similarity search. This is called alignment. Phylogenetics, and then the amino acids, protein structure, and nucleotide structure, or three. So that means there are three practicals covering these three major subjects under me. So the case, I don't know lah. Or the case will teach your second half. So uh, so the practicals will cover these lecture 2 and 3, and then give you the better idea what I'm talking about. All right. So stay tuned. So I will put up the practicals on Monday, next Monday, and you download and try to do it. Okay. So I will give you a lot of guidance, guidelines in the practicals. All right. So maybe you don't understand what is this gap extension, what is the matrix, what is this e-value, this even the alignment of this diagonal line and so on. It's okay. So when we do it, then you know it better lah. So this kind of like manual alignment and using hand and eyes. Yeah. So no computer to help you, but it will be a simple thing to do lah. Okay, guys. Any
Okay,
question?
dokter.
Yeah. So um, it's all from me lah. Okay. So this is my first time to teach this subject. I need to prepare a lot of things. But understanding self-study, when I do my work, I do self-study for these things lah. Okay. But when turn it to teaching material is totally different things lah. Okay. So what I want to say is, give me some time. Sometimes I will be a bit slow to give you the, the, the information or the material for teaching and practical stuff. Okay. All right. Anything you want to say before we end?
No, dokter.
Yeah.
Tidak,
So,
dokter.
okay. Thank you very much. So I end the lecture here lah.
Thank you, dokter, for today.
Okay. Welcome.